Thank you all for coming. I guess that we have a lot of students joining this time. There will be a lot of people that does not know what SONet is, and I will not explain it. I'll let you all Google it and contact us if you're interested in the future. So we'll keep it a little bit mysterious, and then we'll not spend time talking about it. But yeah, I have it here. SONet is the name of our network. Just check it if you are interested in space and future habitats. And with that, I would start. I'm usually introducing the speaker of this SONet Talks we do monthly. This time I'm giving the presentation myself. So my name is Gisela Detrell, and I work at the Institute of Space Systems at the University of Stuttgart. Since a lot of people have joined, I'll just give the reminder that we're recording the session and we'll publish the presentation part online. So if you don't want to appear on the video on YouTube while I'm talking, turn off your camera. We'll have questions at the end. Since we are a lot this time, feel free to start writing questions on the chat if you want. I'll have a look after the talk, and can also just say that you have a question, and we can try that everyone turns their microphone in turns to ask the questions. So then I'll start. As I've just already said, since we have some more people joining, my name is Gisela Detrell, and I work at the Institute of Space Systems in the University of Stuttgart in Germany. And I'm working on a life support system. And that's why today I'm going to be talking about surviving in space. So first of all, what does it mean surviving in space? It's not about being comfortable or feeling nice in space, but having the minimum things that we need to live. And that is for humans, oxygen, water, and food. There are some average values that we can find on the literature. Of course, it varies a lot from one person to the other, but as average values, we would need this less than a kilogram of oxygen, 3.5 liters of water, and 600 grams of dry mass food. We would at the same time produce certain substances during the process and heat, and we would also need some hygiene water. The quantity depends on each one. You probably all know that not everyone takes or you've noticed that not everyone spends the same time on the shower. It's really a personal thing how long hygiene water is required. If we do a quick calculation, what does it mean for a space mission? These quantities, we would see that for 6 astronauts, 500 days, that would be 2.5 tons of oxygen, 2 tons of food, and a lot of water, depending on the hygiene water. But if we put everything together, we come to at the minimum of 30 tons, considering also the tanks that would be required to store that. And that is about, probably 2 to 7 rockets that would be required only to bring the survival things that we require. We still don't have an habitat, we still don't have our clothes and the experiments and everything we would require. That means that we cannot afford that would be too expensive. So what we need to do is take all these substances that we are producing as waste and treat them to make sure we can provide new, fresh oxygen, water, and food. And the way of doing that, this thinking of taking care of the things we produce and reusing them is also something we already do in our daily life. If you plan your holidays, maybe not to Mars, but if you go hiking for a weekend, you take everything with you. You take the food, you take the water maybe as well if it's a short trip. Everything is on you. But if you go out for a long period of time, couple of months on holiday somewhere or exchange student somewhere, you'll take things that you will reuse. You'll reuse your clothes, you're gonna clean them in the place where you're going. You will take some money to get things from the place where you're going, so you will use resources in situ. And that's all very similar to how to design or plan a space mission. So for a short mission, like a trip to the moon, 2 weeks, that's quite a short trip. Astronauts took everything they required with them, and they dispose of all the waste that they produced. If we want to plan a city on Mars, where people can live there permanently, we will need to take all the waste that is produced and recycle that, so we are completely independent from Earth resources. That is quite difficult, but that should be the goal if we plan long-term such a Mars mission. We can have something in between where we recycle partially and we still get some fresh inputs from Earth, and that's what's currently happening at the International Space Station. How can we do this recycling? On Earth, we have biology taking care of that for us. We have plants around us that produce oxygen and extract the CO2. We can also eat the plants. They also contribute to the water cycle. We all have heard since school about the water cycle on Earth. So biology is able to provide everything that we require. But we can also do that using technology. We can use physical or chemical processes. For example, a lot of people in their houses have reverse osmosis for water cleaning. Or we can also produce oxygen by splitting water in oxygen and hydrogen. So there's a possibility outside of the biology to take care of the oxygen and the water, but not to produce food. For that, we need the biology. So for the different types of technologies that we can have, each one is more appropriate or more advantageous for a specific mission duration. So for the very short missions like the moon base, like the moon missions, the 2 weeks, the Apollo missions, an open system bringing everything with us was the best option in terms of the mass that we require, but also what that implies in terms of power, cooling, and so on. But if we want to have missions a little bit longer, some months, we can think now on the International Space Station onwards, physicochemical systems, those able to recycle water and oxygen, start gaining importance in terms of equivalent system mass. So not only the mass, but as I've said, it considers also power and cooling requirements. If we go to very, very long missions, that Martian city that would be inhabited for decades and decades, then a biological system would be the best suited solution. But it's important that you see that in this graphic, the vertical axis, the equivalent system mass, it's actually broken because this green line would probably be a couple of screens over yours right now. So it's farther away up on the vertical axis. But we can also have a combination of these biological technologies and the physicochemical that we know today. So we can have a hybrid system, which starts making sense after duration missions of a lot of months, a few years. So where are we there today? I've already mentioned that on the ISS we have a partially open or partially closed, depending from which perspective you look at it, system. And we have had on the ISS now for over 20 years astronauts living in there permanently. And they were capable of recovering 42% of the oxygen from the CO2 that the astronauts exhale. We're also able to recycle 90% of the water, so not 100% yet, but going on that direction. But we're not able to produce food. There have been some small experiments on the ISS you've probably seen, and I'll mention them later on. But food production has still not been part of the life support system on the ISS. That's how the ISS system looks like, more or less. It's a nice sketch to show all the tasks that are done on the ISS. So there's water recycling, there's the air management system. Food, I'm missing always in this sketch an arrow indicating that the food is coming from outside, but okay. And then there's waste disposal. I'll briefly now go through the different groups or the different tasks and how they are actually carried out on the ISS. So we have from the water recycling, we have urine recovery. So the urine is treated to produce water. And for that we have, for the urine, the so-called vapor compression distillation. So there is a technology that very simply explained evaporates the urine and then it condensates the water so we can obtain pure water on the system. That's a system that is rotating because for phase separation we need gravity, or at least create some artificial gravity. So it's a rotation system that is able to separate then this water vapor so we can obtain this pure water. That's a very complex system and it's moving, so it is not a big component to treat a lot of water per day, a big quantities, but it has been working for several years now on the station. There are other type of waters that are not urine. Urine is quite complicated to process because of the compounds that are in the urine might develop to things that are toxic if not treated properly. But for other, let's say, more simple wastewater, like what is condensated from the atmosphere or water that has been used for hygiene purposes, a multi-filtration system can be used. Multi-filtration is a system with different filters, and I'll not go into the details. It goes from a filter that physically traps particles. So what the first thing that we imagine in a filter, like it has some gaps where the water can go through, but big particles can't. But there are also some more complex filters that take care of different compounds that we can find on the water. And it's quite big. Those are the so-called racks. If you don't know what a rack is, you have to imagine a big wardrobe, like the one I have behind me. So it's quite big compartments to treat water for 6 astronauts that are right now on the station. Besides the water, we have the air management. We have to get rid of the CO2. The CO2, if it keeps accumulating in the atmosphere, it's toxic. When I give this talk in a class, I tell the students if we would seal the class, the doors and the windows right now and staying here for a while, at some point you would all start feeling tired and have a headache because of the CO2 accumulation. Maybe you know it from your lectures when back that times when it was possible to have lectures physically at the university, that you start feeling that the air is I don't know how to describe it, not more dense, but that it's not that nice anymore. And while once you open the windows and get fresh air, you'll get to the regular CO2 levels and everything's good again. So CO2 needs to be extracted. On the ISS, they cannot just open the windows and get fresh air from the outside. So there are different technologies that are taking care of removing the CO2. Those are also some kind of filtering systems or materials that are able to absorb CO2 particles, so the air goes through an absorbent bed. There are different materials. There are zeolite-based materials or amine-based materials, but the ones currently used from the Russian side and the American side are the Bostuk and the Cedra. You can see here a couple of pictures. No, a picture of the Cedra here. Then we have only removed the CO2 from the atmosphere. Then there's the next step of actually gaining the oxygen again from the CO2. And that is the so-called CO2 reduction. And for that, there is the so-called Sabatier reaction. In a Sabatier reactor, the CO2 reacts with hydrogen. And on that process, water is produced and methane. The water can then be used and electrolyzed to produce oxygen, as we will see right now. So it's not the goal is not to obtain water for the sake of having water, but then get oxygen out of the CO2. That has also been installed in the station for a while. It has been down now for a while, but it's coming back. And that's the part then responsible for this 42% regaining of oxygen from the CO2. The oxygen production is done by splitting water. So we can split the water coming from the Sabatier reactor, but also fresh water coming from Earth. It's a way of transporting oxygen with an easy tank. If we have pure oxygen on a tank, the high pressure, then the tank walls need to be very thick and it's very complicated. But if we are able to split the water, if we already have this technology on board, we can transport water and then split it into oxygen and hydrogen. And for that, there's the Russian segment, the technology, the electron, and there's the oxygen generator system from the American side. Here's a picture of the electron on board the station. So those were the American Russian technologies, but we also have European technologies. They came up to the station a couple of years ago. And the main characteristic of the European technology is that it's all compact in one. So it's not that the technologies are very different. We have CO2 removal, in this case amine-based. We also have a Sabatier reactor and water electrolyzer, but it is all together in one rack. So we have one rack that takes care of all and not different technologies separated among the station. It was already built in as one compartment, and it should be able to provide for 3 astronauts. So taking care of the CO2 for 3 astronauts and producing enough oxygen for the astronauts. Although it's important to say that water needs to be added to the system to have enough oxygen in the system. So the last thing is food, and as I've said before, 0% is produced on the station, so it's brought from Earth. And here you can see a couple of pictures of how the food looks like. So it's not magic pills or anything similar, but quite normal food. It is only important that it is processed and packed in a form that can be stored for long periods of time. But it's not something futuristic or crazy. It's something we all may have in our kitchen. Food can be dehydrated, and we have a lot of different fruits dehydrated that we can find on the market. The apple chips or other things in the cereals that are dehydrated. We also have food stored in cans. That's also something that has been done for many years. But again, it's important only that it can survive the launch to space and that it can be stored then for long periods of times. It might also be a little bit modified because taste sense might be a little bit different in space, but it's quite normal food. We had the chance here in Stuttgart a couple of years ago to try some of the food that the German astronaut Alexander Gels had in the station. So we're able to eat some traditional food from here, from the region, stored in a way that it can be used for space applications. And last, the waste on the station. There are no further recycling systems for the rest of the waste that is produced. So it is all stored and disposed on re-entry in the atmosphere. So that's where we are now today. Those are the technologies. I have not mentioned maybe one technology that is being tested for further water recycling, but more or less that is the status that we have today on technologies on the station. But if we want to go farther away to a lunar base or maybe the Martian city that I was mentioning at the beginning, we have to do a little bit more. We need to be more independent from Earth resources than we are today. And that means we can, from one side, improve the physical-chemical system, so increase this 90% water recycling up to 100 or close to 100. We can also increase the 42% oxygen recovery, but with physical-chemical systems, we're limited by the not possibility of producing food. So at some point, we'll need biological components. And that's what I'd like to go for the next minutes, explaining what is being done or what has been done. And I like to start with a quite old example. That is the Biosphere 2. That is a huge building. There's a picture here, I think, of a small car. So you can imagine the size of the building. It has a surface of 16,000 square meters. It's situated in the US, in Arizona. And it was, or it is, it still exists. It's a huge building with different areas from, with plans from different regions of the wall. And it was meant to be inhabited by 8 people that need to be able to survive with what's inside the building. So the building was completely sealed from the exterior. And there were 2 experiments, one lasting 2 years and another one several months. The experiment lasting 2 years had a lot of problems. I will not go into all of them. I'm not the best expert on Biosphere 2, but one worth mentioning is the decrease in oxygen on the atmosphere. So they had to add at some point some oxygen from the exterior because they saw a continuously decreasing oxygen level in the atmosphere, and it would have become a problem for the people inside. So the first thing that someone like me, an engineer, thinks is, if we didn't have enough oxygen, that's surely because of the plants. The plants were not working as they should have. The plants are fault. The real problem was that the materials that were used for the construction of the building were trapping CO2 particles. So there was not enough CO2 on the atmosphere for the plants to produce enough oxygen. So that shows us how quite often engineers like me tend to blame biology for the problems, and the problem has nothing to do with it. So I particularly like to explain that for this. I've said there were many problems, so that also during the 6 months experiment that had to finish because someone sabotaged it by opening the building. So it all sounds very dramatic and bad, like it was a disaster. Well, it was not. There were a lot of lessons learned and a lot of information, biological information on growth and plants that was put together during these experiments. And the most important lesson learned, I would say, is that it is very hard to reproduce our Earth environment in a very small building. This building is huge, but compared to the size of our Earth, it's very small, it's very tiny. And on Earth, we have big buffers. We have a huge atmosphere. We have the oceans that act as a buffer. So when something changes a little bit, it doesn't have a big immediate impact in our planet. And the atmosphere and the oceans serve as a buffer to compensate or to damp a little bit the effects of this problem that might occur. If you have such a small volume, you don't have these buffers. So as soon as something goes out of balance, everything will go wrong. And that's why I like to talk about another project with a very different concept behind, but still biology and closing the loop, providing all the resources without any external interaction. And that is the project, the European project called Melissa. It has been running for 25 years and they plan to work for 24 more at least. So it's an ongoing project taking small steps towards closing the loop. So making sure that from the waste that is produced by the crew, we are able to provide all the resources that we require. And not doing that by putting a lot of biology in one room and hoping that the balance stays, but rather looking at different organisms and making them work independently. So it literally has different compartments with only one inhabitant type on each compartment. So there's compartment 1, 2, and 3, which contain different type of bacteria that are able to go from the waste to obtain nutrients for plants and microalgae. So I'll not go into the details of the bacteria. I'm also not the expert being an engineer, but each step produces one certain substance, and having only one living organism on each compartment, it's very easy to model that and to control that. Same as we control mechanical systems with putting more light or less light, we are able, or they are able to control quite accurately the living organisms. If you have them all together, it's very complex because there are too many interactions that we might not fully understand. And the last compartment is the either higher plants or microalgae, so the organisms that through photosynthesis are able to produce the oxygen and the food for the crew. They have a pilot plant in Barcelona, and they have already tested all these compartments separately. And I haven't checked recently, but the last news I had that they had tested already 1, 2, and I'm not sure if 3 all together working. So they are, yeah, slowly increasing the circle until they can fully close it at some point. This is all designed for Earth applications, so the systems, the technology used is not has not particularly been developed for space applications. So the next step would then be how to apply this loop closing also in space. There are other experiments, and there are many. I'll only mention a couple of them, looking on how to efficiently grow plants, still on Earth conditions, because all those experiments are taking place here on Earth. So we have Earth gravity and other parameters that we cannot change. But one of the ones I particularly like to talk because it's close by here. It's also cooperation within different countries. It's an EU-funded project, but with the German Aerospace Center being part of it. It's Eden ISS. Explain very briefly, it's a huge container. It's actually 2 containers that were modified and sealed to grow plants in the interior, and they were placed or they are still placed in the Antarctica. So they are in an isolated environment and there's no one living in them, but it's close by one of the stations in there. So during a year first, there was one of the German guys in the project living there in the base and going to this station to take care of the plants. It was not a space mission, but I guess it surely felt for him like that, because every time he had to leave the habitat, he did not require a spacesuit, but he required a lot of clothes to put in to get out to the cold weather, and had probably with bad weather sometimes a hard time walking until he could get to this module. And once in the module, he had to take again all the clothes out, so kind of like wearing a spacesuit probably. And here it's about studying how plants grow and how to monitor them and more efficiently cultivate them. So again, it's under Earth conditions, but it's about gaining information on how to cultivate more efficiently. Same with the lunar palace, that's the Chinese experiment module. There have been several experiments. In this case, I think there's screw compartment. I'm sorry, I don't really speak Chinese, so I'm not sure what the words in here mean. If there's someone in the audience that does, I'll be happy to find out what these words mean. But I assume they also have some research lab equipment, and I think a small habitat module, and then the module with the plants. And you can see here, it looks quite similar like the IDEN ISS project. But those are all experiments on Earth. There have been also in space. I mentioned that before, that there have been some small experiments producing food in space. It came on the news several years ago that they had produced the first salad and that the astronauts were very happy to eat the first in-space produced salad. There have been several since. And both Veggie and now the Advanced Plant Habitat Facility are currently in use, and there are a lot of experiments taking place right now with both platforms from the American side. Those are both NASA testing platforms. There are also Russian equivalents, but as you can see, they are all quite small. You see the size of the hand here as a reference. So it's only possible to cultivate very small amounts. So it's not for food producing, but actually to understand how plants grow in microgravity and in different environments. There was also an European system that was used to test plants. That's the EMCS. It's no longer on the station, but I like to show it because the particular thing about this equipment is that it has two different platforms and both have a rotor inside, so this can actually rotate. So it was possible to have in the same module, in the same experiment facility, one system in microgravity and the other one rotating, creating 1G. So one could directly compare an experiment with all similar conditions of radiation, temperature, everything's the same for the two experiments, one in microgravity and the other one in 1G. So we were able to isolate, or they were able to isolate the gravity question for the experiments. That's also interesting if we want to see how plants react to lunar or Martian gravity. So if we have a rotation system, we can actually adapt it, change the velocity of rotation to adapt it to lunar or Martian conditions. So that was all a lot about plants, and plants will surely play an important role in future space missions, but there are other things that could also, and those are microalgae. And I particularly like talking about that because that's the research I do here in Stuttgart. So microalgae are also capable of carrying out photosynthesis. So they are like higher plants. They are actually also some kind of plants. But microalgae in particular are so small that we cannot see them with the naked eye. We require a microscope. I'll show a picture later on. And what we actually see is the green liquid. The algae grow in a liquid in liquid form. The cells swim on the liquid. And microalgae are particularly interesting for space applications because they are very efficient. Efficient in terms of how fast they grow, how much water and light they need, how much space we require for them to grow. And efficiency is a very important word in space design missions because we always have not many resources, we have limited power, so efficiency is very important. The only problem with algae, not the only problem, but compared to plants, the main disadvantage is that because they are mostly protein, we cannot just eat algae. To have a balanced diet, we'll also need to add other things. So we could theoretically eat maximum up to 30% of algae of our daily food consumption. But they're a potential candidate to work one day together with higher plants, not as an alternative, but to work together. There have been experiments already with some algae and some other living organisms, not on the ISS, but in separate spacecraft. So there have been a couple of examples here. I'll not go into details. But I'll go back to only the algae, and I'll briefly explain to conclude this presentation about the experiment we did here in Stuttgart working on algae. So here you can actually see the different algae cells at the microscope, picture from the microscope. And here is the experiment we developed here in Stuttgart, the photobioreactor. So the goal of our experiment was to be connected to the life support rack I've mentioned before, the European technology for air management, to show that the hybrid technology, the hybrid life support system is possible. So the combination of current existing physical-chemical technologies with biology. So here we have the life support rack represented, so the CO2 removal, the Sabatier and the electrolytes that I mentioned before. And our goal was to take a little bit of the CO2 from the astronauts, get it into our algae, and wait for the algae to produce oxygen and biomass that could potentially one day be food for the astronauts. Designing such an experiment has a lot to do with biology, but also with technology. That's why we had a lot of engineers working in the project in parallel with an expert on biology and biotechnology. So there are a lot of things to design. I'll not go into the details, but it is designing such an experiment. It's all about designing a proper lighting system, the reactor's chamber where the algae grow, so the home for the algae, the entire air management inside the experiment and exchange with the outside, because at the end we need to get the CO2 inside and the oxygen outside for the astronauts. And of course, there's a lot of monitoring required, so a lot of different sensors on the process and actuators to make sure that the thermal and air management, it's all under control. We also had some liquid exchange ports, so we could insert nutrients. The algae also need their own food, so they take nutrients from time to time, and then also extract at the same time the produced biomass that could potentially be food. That's just a picture of the reactor chamber we had inside. I'll not go into the details, but you see here some channels, and that's where the algae would be moving around. Just a couple of pictures of the experiments we had on Earth. So that's how the algae looked at the end, looked quite dense. We let the algae grow a lot in the reactor, so we got at the end a very dense mass. We had a membrane for the gas exchange where we could see some accumulation of the algae after having the experiment run for over 180 days in our lab. And that's how the algae can look like. So we can have nicely algae cells swimming freely, not really swimming, but letting be moved. They don't actively move themselves, but on the liquid, we can also see agglomeration clusters forming from time to time. We can also see cells getting damaged because of the pump that we have on the system. There might be stresses on the system, so that can also occur. And what we're interested as engineers, and I'll not go into the details, but was to see how the algae were growing, to see how the cell density was evolving inside the experiment. We saw that for the experiments we did on Earth. Those are two experiments that took place on Earth before we sent the experiment to space. So from one side, how the algae grow, and we can see that we clearly saw a grow on the system. And then we're interested to see how the CO2 is being consumed, so how much is being consumed and how much oxygen is being produced. And we saw that the system, it was working. We can argue how efficiently, but we proved the feasibility of the system. And then we sent it to space. So we had our algae prepared here in Stuttgart sent to space. The experiment was installed. And after 2 weeks, we were able to take a nice picture of the algae. So that's a sample in one of the syringes that we use for this exchange, where we could see the algae getting out of the reactor after 2 weeks looked nicely green as we expected. But unfortunately, we had a power lost after some hours after this sample. So our experiment stopped getting any light or gas exchange or anything that required electricity. So the experiment had to stop only 2 weeks after we started. We planned to work for 6 months, so it was strongly reduced. But we saw that the algae were growing during those 2 weeks, so the biology itself was working, even if we had some technical problems during these 2 weeks, but mostly then with the power loss at the end. So once again, a similar message like with the biosphere too, that we tend to be worried about the biology, being it complex and difficult to handle, but usually when we had problems, it tends to be because of the technology. And the algae actually adapt themselves quite good. So it was, even with the problems, the algae kept growing inside the experiment for those 2 weeks. So biology always finds its way. We got some samples to analyze, but of course, being the experiment shorter than expected, our results were also more limited, but we are looking forward to continue working on that area. And with that, I've reached the end of my presentation, and maybe another important message that everything that I've explained has to do, of course, with the life support system of the future. So how could it be in a future space base somewhere? But it also has to do with what we do here on Earth. Algae can also be used to reduce CO2 levels here on Earth, to treat water, to produce a certain amount of food with very less resources. So we have to always keep in mind when we design things for space that they might also contribute to help us solve problems here on Earth. So always keep both things in mind, the space side, but also the Earth application side. And with that, I get to the end of my presentation.